Good morning. I would like to welcome everyone to the 2018 Texas A&M Plant Breeding Symposium, the Omics Era, a new page in plant breeding, both here on campus and live via our streaming webinar. My name is Amani Kianam, and I'm a PhD student in plant breeding and the chair of the organizing committee. I would like to start by thanking my fellow organizing committee members, Yuya, Natalia, Steve, Lauren, Daman, Nolan, Blake, and Anil. This group of individuals has helped make today's event possible. They are frantically working in the background to make sure everything is in order, and I hope you get a chance to meet with them during our breaks or during the networking social tonight. The Texas A&M Plant Breeding Symposium is part of the DuPont Plant Science Symposia series. The goal of the symposia are to enhance discussion amongst the academic community and private industry, build key organizational and management skills in future plant scientists, and expose students to career opportunities in agriculture. The event is exclusively run by graduate students studying plant breeding and related sciences under the guidance of Dr. Tabre Abadi, Senior Research Manager for DuPont Pioneer, and Dr. Jason Rauscher, Academics Relation Manager. Dr. Rauscher is attending today's event along with two other Pioneer representatives, Drs. John Van Hammert and Dr. Heriberto Torres. I encourage students and faculty to speak with these industry representatives during one of the breaks today. As our main sponsor, DuPont Pioneer has made significant contributions to Texas A&M Plant Breeding Symposium. Please join me in thanking DuPont Pioneer for their continued support. We have also received support from several on-campus departments, including the Departments of Soil and Crop Sciences, Institute for Genome Science and Society, Biology, Horticulture, Molecular and Environmental Plant Sciences, Biological and Agricultural Engineering, and Plant Pathology. We have also received support this year from the College of Science and Engineering of the Texas A&M System's Corpus Christi location. We've also received a generous contribution from the Texas Peanut Producers Board. Many of our attendees have received complimentary symposium t-shirts today, which were generously supplied by Cotton Incorporated. We also received beverages from Pepsi Cola. Ladies and gentlemen, please join me in thanking the rest of our sponsors. We invite everyone to follow Texas A&M Plant Breeding Symposium on social media and tag us on your posts today. We'd like to know if you all are having a good time. We are committed to enhancing networking between our speakers, sponsors, students, and everyone in attendance. Later tonight, after the symposium, we will be hosting a social at Cafe Excel. Everyone is welcome to attend, including your significant others. There will be free appetizers and a cash bar. The symposium today is also hosting the 4th annual Graduate Student Research Competition, which includes rapid oral presentations and a poster competition. This competition promotes and encourages students to communicate their research to their peers, as well as network with a wider plant science community. From Texas A&M University, Anna Costeau, Nicholas Ace Pugh, and Mahendra Bhandari have been selected from a pool of applicants to give a rapid oral presentation in the second session today. In addition, they will present scientific posters of their work during the following poster session. They will each receive a $250 scholarship for their effort. At 11:40 today, there will be a 1-hour poster session as a part of the competition. Poster competitors were room by a panel of plant science experts. First, second, and third place winners will be awarded $200, $150, and $100 respectively. We encourage all poster presenters to stand by their posters during the poster session. The DuPont Pioneer Travel Scholarship was created to foster interaction between graduate students studying plant breeding or a related science at Texas A&M and other institutions. This scholarship, administered by the organizing committee, is fully funded by DuPont Pioneer. Katie Rogers, a PhD student from University of Florida, and Suraj Sapkota, a PhD student from University of Georgia, will each receive a travel scholarship of $450. We will be hearing from them in the second session as well. We thank each of these student researchers for joining us today. Please join me in congratulating all these student research competition participants. As stated earlier, the Texas A&M Plant Breeding Symposium is part of the larger network of symposia called the DuPont Plant Science Symposia Series. These symposia are completely student-run and are held at major plant breeding universities across the United States and throughout the world. We are thankful to host one of these events on our campus for the 4th consecutive year. Excuse me. It would not have been possible to bring these world-renowned scientists from across the country to College Station if it weren't for the significant contributions of DuPont Pioneer. To learn more about DuPont and their network of symposia, we will now hear from Dr. Jason Rauscher. Dr. Rauscher received a PhD in evolutionary and population biology from Washington University in St. Louis. He received postdoctoral training from Cornell University with expertise in genetics of plant adaptation, diversification, and polyploidy. He has over 20 years of experience in academia and research, including time as a Fulbright Research and Teaching Scholar in Columbia, an NSF International Research Fellow, and a Research Fellow at the University of Puerto Rico. Please join me in welcoming Dr. Rauscher.
Thank you, Mani. My mic is working. Well, the Ag Division of Dow DuPont, depending on which you prefer at the moment. I'd like to welcome you all to this event, and I'd like to, thank you, take the opportunity here to tell you a little bit more about the series, societal economic impacts, and quite frankly, I'm sure most of you know somebody maybe in the room that is a farmer, and you see people have to deal with this. And so the question is, what can we do to kind of, alleviate, address this problem? And so for today, I'll be talking about cotton, but really you can pretty much insert your favorite crop here in terms of the technology and the applications. And so this is where cotton is produced in the U.S. We're in Texas, which you probably already know is the largest cotton-producing state, so this is always good to be in Texas to give this talk. But in terms of where cotton is produced, it's produced along the southern part of the U.S. This is affectionately known as the Cotton Belt. And it has the unfortunate realization this is where a lot of heat and drought stress occurs in the U.S. And so in any cotton-growing region, you're typically going to experience these adverse environmental conditions. And so 2011 here in the state of Texas, you guys had one of the worst droughts ever. $2.2 billion economic loss in cotton production alone. You guys lost $5.5 billion in cattle. And so additionally, 55% of the acres planted that year were just abandoned in the field. No attempt to even, save the crop at all. And unfortunately, global data supports the same pattern in other cotton-producing regions, whether it's China or India, that this is a continued problem that will be facing cotton breeders as well as cotton production, primarily, going forward in the future. And so the challenge is, how do we develop heat and drought stress tolerant varieties? Because unlike our, I can guarantee every one of you in the room is wearing a piece of cotton. Whereas, the same thing can't be said for what you had for breakfast, whether it was one of the major cereal crops. And although, cotton is not food, and I'm not going to stand up here and tout the benefits of fighting, world nudity, the fact is there is no economically sustainable solution for this fiber crop. And why, this is why, there's so much emphasis on how do we sustainably produce this fiber. And so really, heat and drought stress tolerant varieties is plant breeding. The drought phenotype is a whole plant response. And it's a phenotype that really relies on the environment in which it's expressed. And because of this, I would argue, and I think the literature really bears this out, that there really is no single gene solution. And primarily we look at, there's so many components. I mean, in terms of photoprotection, we have the actual leaf architecture and the composition in terms of the pigments that make up the leaves.
At the other aspect, there's transpirational efficiency. How water is being pulled through the plant, and the trade-off that the plant must make in terms of losing water to fix carbon, and how vapor pressure deficit really drives that, demand. There's also water uptake, how the roots are exploring the ground to capture water, how it's being driven up into the plant itself, and during times of drought, how that root architecture explores deeper other areas to acquire that water that's needed. And finally, there's always the basic principle of partitioning. In terms of bulb production, leaf production, stems, roots. And so because of that, plant improvement really has to occur at the whole plant level. And to do that, we really need to understand the underlying biology as well as the genetics of stress response. And although primarily I focus on transpirational efficiency and water uptake kind of in my research, it's really all these components together that, give a plant its drought resiliency. And so that's kind of the challenge we're facing now, is how to integrate physiology and genetics to really meet the demands of the plant as well as, future crop plant production areas. And so kind of illustrate this at another level is that when we think about genotypic information, it's really bit information. It occurs in two dimensions. It's A's, T's, C's, and G's. And, for the past 20 years, we've gotten really good at, finding these, cataloging them, storing the data. But really, it is just a two-dimensional space. And it's through the interaction with the environment that we add this complexity where you can see finally in this final figure, we get these, phenotypic optimums, just like any selection paradigm, where there's an optimal phenotype for different environments. And it's only, through this interaction of the genes with the environment that these arise. And so the challenge is how do we, find these optimal phenotypes for the different environmental challenges. Questions that guided this research, first from an engineering standpoint is it possible to actually collect meaningful, physiologically relevant data at field scale under field conditions? In terms of more of the applied breeding, is there exploitable variation for stress adaptive traits in cotton? We all know that you have to have variation. That variation must be heritable. So does that exist? More just a basic genetics question is there a temporal basis of QTL expression patterns in cotton? This idea of when genes are turned on in relation to the environment as well as actual plant development. And then finally, very applied, can we use physiological trait data to actually predict agronomic performance? So obviously I would make the argument that high-throughput phenotyping is an essential tool and that it gives us the ability to evaluate large populations under field conditions. And this is imperative for drought research because there's such a dynamic interaction between the environment and the plant that, soil crusting, hardening, you can't really replicate those exact same conditions in a greenhouse environment. Also, typically the results you see in a greenhouse don't generally translate to the field environment as well. So that's why having this field component is so essential. Additionally, we're able to measure the trait throughout development. Instead of just taking these static time point measurements and only seeing really what's happening at that one point, by longitudinally assessing the phenotype, we actually get more of a video of what's going on, how the trait's developing in relation to phenology as well as the environment. From a research standpoint, we can work with larger genetic populations to increase power for genetic mapping, GWAS, genomic selection as well. And I would also argue that there's a lower cost and minimized subjectivity. As field breeders are walking through, you're scoring plots as fast as you can because you need to move on to the next one. Because of that, we're typically bias ourselves towards visually scored traits. Lower cost in the sense that the upfront buying the instrument can be hefty, but once you have it, typically it can run for many years. And I don't want to knock the undergraduates, but machines don't get hung over. They don't complain about the heat. So it reduces that layer of complexity in the data as well. And so the work I'll be talking about today was the work that was done in Maricopa, Arizona, in conjunction between the University of Arizona as well as the USDA, and kind of their pioneering phenotyping work using this platform that you see in the photo. And so Maricopa, Arizona is really an ideal place to do this type of work. We have what can be considered extremely consistent environment. It's always clear skies, it's always hot, it rarely rains, and this makes for really, beneficial place to do this type of research. And so in that graph on the right, you can see that red line is 32 degrees C. Above 32 degrees C is when cotton really suffers a loss of efficiency in terms of Robisco-fixing carbon. And so in these 3 years that this trial was run, there's only 10 days where the temperature does not go above 32. And so because of that, we have this nice consistent environment. The other key component is this managed stress. There's a precision irrigation system, there's a few of them, allowing us to really finely control how much water the plant's receiving. And for this work, the water limited treatment as well as well watered. The water limited basically received 50% of the daily evapotranspiration rate of the well watered. And this was applied using subsurface drip irrigation. And just to note that the irrigation treatment itself was not started until 50% of the plots had already begun to flower to reduce the effects of, resources for real mapping. And it just so happened that TM1 became the reference genome that was recently sequenced. Just the basic field design, alpha lattice, the plots were, 9 meters long. So we had a good sample size. You can see how they're arranged in the field in terms of staggering wet, dry. And the phenotypes that we're looking at for the canopy are temperature, NDVI, and height, which I'll describe shortly. Physiological trait data, abscisic acid content, and carbon isotope discrimination, 2 hallmark traits for drought phenotyping. And then, additionally, we looked at agronomic traits like lint yield, boll size, as well as fiber quality, and then some seed ionomics data, which I'll talk about in the second part of the presentation. And so here's the actual platform that was used. It's not nearly as sexy as some of the things that have been designed recently, but this was developed in 2010, and kind of just a basic set of sensors. Active multispectral canopy sensor, the crop circle, ACS 470, which some of you may be familiar with, which was measuring canopy reflectance. Infrared thermometer, as well as a sonar to get canopy height. We had onboard data loggers to collect everything, provide the georeference and timestamp. And then finally, the one piece that really makes this all possible is this GPS RTK system. Standard GPS has a resolution of about 1 meter, which you can see in this photo. 1 meter either way might put you in a different row, or i.e. a plot. And so with the RTK system, you actually achieve that sub-centimeter level precision, which allows us to tie each data point back to a specific point in the field, i.e. a plant. And so with this platform, it cruises along about 3 kilometers an hour. This translates into phenotyping about 450 plots per hour, which is pretty good. I don't care how many undergraduates you did hire. I think you'd have a hard time beating that efficiency. And so again, just kind of to illustrate this, it's what we're really trying to capture is a longitudinal assessment of phenotypes over time, how these traits are developing in the growing season. But it's also critical to remember that what we're measuring is the plant's response to the stress, not the stress itself. And because of that, that is why it's so critical to have a managed environment, i.e. that we know that these plants, the only thing that they were suffering from was a lack of water. It wasn't like a nutrient, it wasn't biotic stress. And why this is important is for traits like canopy temperature. Canopy temperature is just like when you go to the doctor and they take your temperature. They either tell you that you have the flu or you might die tomorrow. I mean, it's not really indicative of anything. And so that's why it's critical to know what's going on in the field so you can understand, what that phenotypic information is telling you. And so in this case for canopy temperature, it was the plant-water relations. We also used NDVI. Most of you are probably familiar with NDVI as a quantification of biomass. However, in this context, we're using it to actually measure the canopy architecture in the sense that, when plants are well watered, they have full turbidity, the leaves are actually the incoming light. But as they lose water content, you can see in this photo they begin to droop. Because of this, it's not reflecting as much light. What we're trying to do is actually capture that signal using NDVI. And then finally, we have canopy height and leaf area index, which kind of gives us an idea of the whole plant response to heat and drought. And you can see in the bottom that, again, the data collection was initiated at flowering. That's when the drought stress was imposed, and was continued pretty much up until harvest. So here's what the actual geoprocess canopy temperature data looks like. So each circle represents one canopy temperature data point, and you can see that blue means it's cooler, green means it's a warmer temperature.
And so the first thing you can see is obviously the differences between the well-watered and water-limited treatments. But also you begin to see some of the fine-scale differences between the genotypes themselves, as well as the field environment, though. You'll notice that down here in this part of the field, even though this was a water-limited treatment, it's quite blue, and that's because that part of the field had a high clay content, i.e. water holding capacity. So those plants weren't as stressed as we initially thought they were. But again, overall, this gives us a very fine-scale view of what's occurring in the field as well as the genotypic differences. So as all the graduate students know, that to breed, you have to have variation for that trait, it must be heritable. And so that is one of the first tenets and basic questions we had to answer. And for canopy temperature, we saw, very nice distribution. I do want to point out that the temperatures reported, these are relative, they're not absolute. If the plants are at 45 to 50 degrees centigrade, they'd practically be on fire. So just as a note. But you also notice that the heritabilities were quite high, 0.83, 0.9. So this gave us a good idea that we had, fairly good experimental control. We're collecting good data. But what we're really interested in and why phenotyping itself was cool is that we could collect this data at multiple time points throughout the day, 7, 10 a.m., 1 p.m., and 3. And so if we start at canopy height, you pretty much see the phenotype that you would expect, that plants don't get taller or shorter across the course of the day in response to drought. As we move up to the more responsive traits like leaf area index, you'll see there's a slight decrease over the course of the day if you have like a really good eagle eye. But if you look at NDVI, you'll see from 7 to 10, things are doing okay, but it's that 10 a.m. to 1 p.m., that's when the plant, begins to wilt. And that's that change in architecture I was talking about when the leaves start to droop. So we're actually able to quantify that. And then also, if you look at the well-watered, you'll see that actually NDVI slowly increases over the day, and this is from the actual diheliotropism, when the leaves are actually tracking the movement of the sun across the sky. And then finally, you look at canopy temperature. This has been a hallmark graph kind of produced numerous times before. At 7 a.m., things are pretty much isothermal, but by 1 p.m., you can see that the water-limited treatment's 10 degrees warmer than the well-watered. Now, the critical thing to point out is it's not the point measurements themselves that we care about. That's not the phenotype. The phenotype of interest, and what's really intriguing, is that for the first time, we can get the change over time. That slope, basically. And what this represents is the plant's dynamic response to the environment. And this is why high throughput phenotyping is becoming, allowing us to answer a more complex question is we want the plant's response to the environment. That's what we're interested in. And so by having this technology, we could use this as a new phenotype for our QTL mapping. And so for the rest of the talk, just for sake of brevity, I'm just going to focus on canopy temperature. And so given that previous graph, the question was, well, maybe that was just like a standout day, that we got really good data. It was extremely hot. Vapor pressure deficit was very high. But sure enough, when we looked at all the other time points in the season, irrespective of the stage that the plant was in terms of development, whether it was flowering, bull fill, or fiber development, we saw that same dynamic response of canopy temperature in the irrigation regimes. So that was very positive, that we're collecting good data that's hopefully capturing some biological signal. When we move on, we look at the canopy temperature readings themselves, and what became apparent was that they're more correlated within plant growth stages than across, because the idea would be that surely if there was no signal, it would just be linearly correlated across the season, i.e. due to proximity of the measurements. But what we saw was very distinct, groupings that pretty much mapped up with the developmental stages of the plant. This was exciting because it gave us, first indication that the platform was collecting some type of data that had real signal in it that was capturing the biology. It also really illustrates the point of why continued phenotyping throughout the season is needed. If you'd only picked one time point, you would have missed pretty much the entire story and not really capture what's going on in the field. And so using this was, kind of good validation and gave us hopes for the actual QTL mapping. Now, I recognize this figure is a little overwhelming, but let me just walk you through it. So the two panels, on the top is the dry treatment, on the bottom is the wet, the well-watered. On the right-hand side are the 3 years in which data was collected. The left-hand axis are the individual days. And so again, those individual days, we're getting that slope across, over time as the phenotype. And then on the bottom are the actual individual chromosomes with the genetic markers. If you see a red smudge, I guess, those are alleles that are increasing the canopy temperature. If you see blue, that means it's decreasing it. And so the first thing we found was that we could detect specific QTL that had a very defined temporal expression. That these, were detected at the same time of the year in the 3 years. So this was super exciting. This was the first time we had empirical evidence supporting what we all thought in terms of, we know that genes just aren't turned on at the beginning of the plant's life and left on. That there is this coordinated expression, whether it's, due to developmental or whether it's in response to the environment, that there is, controlled expression of these genes. The other thing that's important to remember, though, is, we're in this age where sensors are cheap, generating data is easy. Everybody can do it. The question we have to keep in mind is the data that we're generating biologically relevant? Is there something supporting, what we're seeing? And so one of my concerns was that we're maybe just mapping noise, that we were duped by our own machine. And so those other traits I was telling you about in terms of ABA and carbon isotope discrimination, the QTL for those mapped to the exact same spots as those for canopy temperature. This was very encouraging to have, validated phenotypes basically serving to say that, yes, what this platform is capturing is relevant in terms of canopy temperature and the plant-water relations. There's that saying, I'd rather be, lucky than good. And 2 weeks after I finished this analysis, the cotton genome was published. And so when we took these QTL intervals and blasted them, sure enough for the ABA, we found an abscisic acid response binding element factor, which was excellent news. And then for carbon isotope discrimination, same thing, a drought response family protein. So again, this really serves as validation that there is biology underlying the data that we're collecting. And I think as we move forward, in terms of all the sensors that are being developed and high throughput phenotyping technologies that come up, it's always important to kind of keep that context in mind. So finally, the one question was, well, if this data is capturing biology, can we use it to actually predict traits that we're interested in? And so lint yield. And so we used all this canopy temperature data in 2012 to try to predict lint yield. And our predictions, I would argue, were fairly good for just using phenotypic data. There is no marker data used in these analyses. And so on the dry treatment, we reached a correlation of 0.7, which wasn't bad, but it raises the question again is, what's that actually mean? Like what is driving that relationship? What is the underlying biology? And so we go back and begin to kind of tease this apart. The first thing we did was look at the 4 individual traits in terms of canopy temperature and DVI, and we used random forest regression to kind of pull out which factors were the most important ones driving this response. And so what we found was that sure enough, canopy temperature was the overwhelming predictor that was the best. Next, we look at the time in the season, those individual days, and you can see days 201 and 208 are vastly more important than the other, and this is when flowering is occurring. And intuitively, this makes sense. I mean, you can't have a cotton boll form without a successful fertilization event. And we know that pollen is liable to heat stress. And so, again, this was just kind of important information for us to really, convince ourselves that what we're doing had some valid meaning. And then finally, when you look at the actual time points within a day, you see that 10 a.m. is just a little bit higher in terms of its predictive accuracy or what's, and why we think that is because at 10 a.m., you're starting to see good stress symptoms in the field, whereas the 1 and 3, I mean, it's so severe that the plants are just kind of blowing out. So at the 10 a.m., we're kind of getting that more fine-tuned response. But the biggest thing is that canopy temperature at flowering peak bloom is the most predictive of lint yield. And this type of information from the producer standpoint can be used to kind of monitor and schedule how they're going to use their irrigation. Say if you only have a certain amount of acre feet that you can apply per year or so, you'd probably want to use that during flowering. So now I'd kind of like to change gears and talk more about understanding environmental effects. We know that the phenotype is relying on the genotype, the environment, and their interaction. But pretty much since the dawn of man and plant breeding, we've done a really good job of phenotyping half the plant. And obviously that's the top half. In-field root phenotyping is in its infancy. I don't know how many people went to phenome last week. There's a few technologies that are developing, but nothing at field scale that you could really employ. But that still doesn't answer the question and doesn't resolve anything. We have to figure out how to understand how the root system is interacting with the soil environment, because as that photo shows, and what we already know is that it's highly heterogeneous. There's a lot going on down there. And we, have not by choice, but because of technical limitations, have kind of neglected that. And so one way we thought about approaching was this: is we could have some type of proxy for the soil environment and include that in our analyses. And so for that, we turn to ionomics, which is the rapid profiling of elemental composition of samples. And in terms of biological samples, elements are an essential component of every cell. And specifically, like for plants, chlorophyll, magnesium is hanging out in the middle of that antenna, collecting all that light energy. And so because of that, changes in elemental concentration, due to abiotic stress like drought or soil composition, can be quantified. And so this, ionomics data can actually provide us information on the overall health and function of the plant itself. And you can see, in this element, the periodic table, which elements we're phenotyping. The green boxes are the essential ones for growth. Purple will kind of trace. And so I guess just to highlight again that this type of information gives us more insight into the physiology of the plant than maybe we were, hoping for. And so in this work, we also performed ionomic analyses on the cotton seeds themselves, but we also did soil sampling throughout the field and analyzed that as well. And so just, again, we know that the soil environment is highly heterogeneous. And so here is the profile of the field and where the real population was grown. And you can see there is quite a bit. In our case, we had the distinct advantage of using the same field for 3 years in a row. And so if you were to think about this in the context of multiple locations, different fields, the impact of the soil environment, that complexity would ultimately impact your phenotype. And so looking at magnesium, one of the key components of chlorophyll as well as other physiological processes, we can see that there is, in fact, this gradient across the field. We used kriging to actually develop this interpolated field map. And by doing that, we can go back and add plot level data for the magnesium concentration into our analyses. And so sure enough, when we did the analysis, we found that the magnesium concentration within the soil was highly correlated with what we saw in the seed. And so by being able to, A, know about that variation, B, control for it, we better serve to improve our genetic mapping as well as understand the biology in terms of how magnesium is used by the plant itself. And so going forward, once we've kind of controlled for the soil environment, was to look at the actual ionome itself. And these are the genotypic correlations. These are not the phenotype correlations. And what we saw is that we got this really strong clustering right in the middle of this network diagram, and that the biologically relevant elements were all grouped pretty closely together. They all had high correlation values. And then finally we had the micronutrients kind of on the perimeter. And you also see that potassium's hanging out there at the top. And we think the reason why potassium doesn't group with anything is because it's so prevalent in just about every process in the plant that there was no real, established relationship. But basically, this gave us our first indication that this ionome, that this biological system is highly, interrelated and integrated. And you can see that between the two irrigation treatments, there's no real differences, i.e. that the ionome responds basically as a unit. And so with that in mind, we had to think about how best to map this trait. And so, as basic geneticists and everybody, you take your traits and you analyze them one by one. And so we did that first. We used a Bayesian classification technique so we could fit all the markers at once to really control for the background effects, because we were concerned about that given the correlation among the traits. And so you can see for the various elements, we've got multiple hits across irrigation regimes. But what we really thought was, well, if everything's so interrelated, we need to take that into account in our analyses for mapping. And so we turned to using a multivariate approach. And so the first thing we had to do was to decide how to group the elements themselves. And so based on the literature as well as some clustering, hierarchical clustering done using the genetic relationships, we broke things down into those elements that are involved in ionic reactions as well as redox. And so we use those as the group phenotypes and perform the mapping. And so the multivariate QTL mapping, detected the same, QTL that the individual elements map to. But more importantly, we were able to detect 9 more QTL that, at a significant threshold, that in the Bayesian analysis, although detected, weren't declared significant. So this shows us that by borrowing information through the correlated structure of multivariate analyses, we're able to improve our power to identify loci controlling this trait, as well as pleiotropic QTL, which in the context of the ionome makes better sense given how everything is so related. But again, it was kind of the same question that we had with, the canopy temperature data was, if this data is capturing physiology, surely it should have some predictive value, that we don't always have to rely on marker information. And so we took the 3 individual years and generated blues for those. We did the PCA and colored things accordingly to well water and water limited. And if you look at a 3D plot of this, it actually gets a very nice separation. So based on that, we thought, well, surely, if we use some supervised machine learning techniques that we could probably predict, the stress level, i.e. if it was water, well watered or water limited. And so using a host of like k-nearest neighbors, linear discriminant analysis, logistic regression, and support vector machines, we were actually able to, do cross-year prediction, use 2010 and 11 to predict 2012. And you'll see that the accuracy is surprisingly high, 97%. And I realize that this is a 2 classification problem, which is one of the simplest ones. But again, relying on, data from individual years in a field environment with no marker data, I would argue that this prediction accuracy was quite higher than I thought we would achieve. And so again, this just kind of harkens back to, I guess the theme is that it's important to understand what the data is telling us, what the biology is, that just collecting data in and of itself is not really, advancing science so much. We have to think about this in the terms of our hypotheses and questions that we're asking. And so just kind of summarize with the field-based experiment, we had this novel platform that was able to measure dynamic traits under actual field conditions, gave us this kind of first insight into temporal QTL expression, and provided us some indication of yield prediction. With the ionomics work, it was this ability to integrate environmental effects into our analyses and better understand how the environment regulates and drives the phenotype itself, and as well as the shared genetic basis and ability to predict stress level. And so now I'd kind of like to spend the last 10 minutes of my presentation just kind of describing some of the future directions, not only my research, but kind of as I think the field is going. And so one thing that's been hampering us is that we've focused on traits at the macro scale level because, that's what technology really enabled us to do, in terms of measuring plant height, yield, flowering time. But really, the intrinsic traits that give rise to those occur at a much finer scale. And so the question is, how do we get at those? And so there's this idea of implantive phenotyping. And so we've developed this nanoparticle sensor that's actually taken up by the leaf through the stomata and resides in the intracellular air spaces. And it fluoresces based on the vapor pressure deficit inside the leaf. And this is with a group out of Cornell, Abraham Struck. And you can see here's a corn leaf. The circled area is where this, fluid with the nanoparticles has been injected into the actual maize leaf. So it sits there for a few minutes to equilibrate. And then when we look at it with the microscope, we get a very nice fluorescence pattern. And depending on the intensity of that fluorescence, we can actually quantify what the inside, what the relative water content is of that leaf. And so it's getting at these, very fine scale traits, I think is one of the areas that people are rapidly progressing. I know there's a group in Iowa State that has these wearable plant sensors, and they're kind of pushing the exact same thing, is that to understand the large scale traits, we need to look at the micro scale traits. The other one I would argue is whole plant imaging. I think this is already being done here at Texas, but one of the tenets of biology is that structure equals function. Nature is very good at designing the things that it needs to be successful. In this case, structure equals function equals a phenotype. And you can see this, I think this is actually a photo from the Texas Historical Society of this cotton plant. And you get this very distinct arrangement of cotton bolls on the plants. And recent work has shown that, the geometrical arrangement of plant parts on, the plant itself, it's not happenstance. That there is some kind of biological mechanism driving that. And so one of the questions is, can we use image analysis to really start to tease this apart? And specifically, the discovery of latent variables. That, as humans, the phenotype concept is very limited in terms of what we think of, what we define as a phenotype. And that actually may be a limiting factor because we haven't considered what other phenotypes might be prevalent that we're just not aware of. There was a group out of China that had an excellent piece of research that took images of plants, extracted over 400 phenotypes for drought stress, and the mapping results from that were far superior to just looking at standard traits like canopy temperature. So surely there is value. I mean, again, though, the other side of that is that as scientists, we want to know why. We want to know why we're seeing what we are. So although these black box approaches can be beneficial, it's always important to keep in context of what they mean. And so with this in mind, I was in New York at the time, and when you're in New York and the closest cotton plant is 2,500 miles away, and it's always winter in New York, you had to come up with basically a model. And so with a 6-pack of beer and some dowels and some cotton balls, I came up with this cotton plant with the idea of, can we image it and begin to do this type of work? And so I just used my iPhone, and I don't think I can play the video from up here. I think you guys have to. Okay, we can maybe just go to the next. Oh, perfect. And so anyway, this is just using an iPhone, very basic technology, stitching the images together. We get a fairly good representation of this model plant. And there was 28 cotton bolls on the plant, and all 20 are successfully, recreated in this 3D model. And what's exciting is that, we can begin to see the branching structure of the plant itself, or of my model plant, as well as the distribution of these bolls. And so that was all fine and dandy, but I mean, that's like best case scenario. That's, sitting in your basement with an iPhone. And so the question was, can we do that with real plants? And so if you could play the next one. And so you'll see that you can actually identify closed bowls in this plant. There's, open bowls as well. There's also cotton flowers that you'll see right here kind of towards the top. We get actual leaf architecture and arrangement. And this is what gets really exciting, is understanding how the plant itself is formed to solve these challenges of resource capture and development. It's a very competitive world out there, and for plants too. And this idea that you need to be better than everybody else equally holds true. And so these different architecture and arrangements of structures become kind of critical in understanding the biology. And so the work I'm doing with like Ian Small and James Kilhesse is kind of how do we scale this up to image entire cotton fields. And so that's an ongoing battle. Finally, I'll just make a quick pitch for envirotyping, that we're spending all this time phenotyping and genotyping to these exhaustive depths, but what do we do with our actual environmental data? There's cheap technology right now to capture all this information, and I recognize that I haven't figured it out either in terms of how you integrate this into your actual analysis. I mean, it's overwhelming, right? I mean, you have not only the field site level, but you can go break it down the plot level. But that's the thing, is that we know that these micro environment factors impact the phenotype. I mean, our ionomics showed that. So surely when we look at, position in the field in terms of relative humidity, these factors all combine to create the phenotype. And so, I just basically making this pitch that I'm sure there's smarter minds than mine out there to actually begin to tease apart these questions and how we integrate this information. And kind of why this is important is it leads to this idea of developmental quantitative genetics. And it's that it's connecting gene expression to actual phenotypic expression over the course of the season. So plants reach a final phenotype, by various methods. Not everybody has the same growth rate. Not everybody, it's just like humans. We don't all progress at the same rate. And how we reach our final phenotype, there might be interesting patterns in that information itself, that development. But more importantly, it's figuring out how that developmental time series happens in the context of environmental variation like precipitation and temperature. And furthermore, tying in the genetic level of when those QTL are expressed relative to the plant development as well as the environment. And so this becomes the hallmark of precision breeding. It's about the idea of having the right gene on at the right time in the right environment. And this is really what's capitalizing on in terms of, G by E and how we maximize that. And so to really carry this work forward, it becomes the idea of data analysis to really connect all these different sources together as well as that environmental data. And so one way that I think a lot of people are thinking about doing this is integration through crop model or ecophysiological models. Is that we're really good at collecting environmental parameters. With high throughput phenotyping, we're now getting some of the traits that we're interested in terms of like leaf initiation rate, canopy temperature, chlorophyll fluorescence, and final phenotypes like yield and quality. And it's the parameters from the models themselves, these light use efficiency. These are the traits I think we're all really after. This is what defines yield. This is what gives rise to the plant itself. And so I think these are really where we're trying to push to, because we know that crop growth is not an additive process. But yet, all of our statistical models are largely additive. And so how are we really going to break that apart if the tool we're using isn't designed to incorporate that complexity? And so some of the work is using this for improvement of genomic selection capabilities and predicting in new environments. And so I know I've talked a lot, but here's kind of like how all this information is integrated together, that we have the genotype, G by E, and environment at the top of the figure. Genotypic information, I would say, is a slam dunk at this point. We're really good at collecting this, storing it, analyzing it. Those tools are available. Our genomic selection models are fairly robust. GWAS has been really worked out. Envirotyping, you see on the other side of the figure, I would say we're largely pretty good at this as well. It's just breaking this down to a smaller scale. And then we have the phenotype itself, and you'll see underneath there's that bang symbol with field-based high phenotype. This is really what's going to allow us to connect the two together. And crop models really offer us a way to integrate all this information. And you can see that using the model parameters from the crop models as input into our genomic models can give us, that deeper insight into these actual biological traits that are driving the phenotypes that we observe in the field. And so, again, combining all this information, it's the ability, like in plant breeding, what's one of the limiting factors is the number of sites that you can evaluate your genotypes at. It's always a challenge. It's the most costly part. But what if you could model, if you had historical data and the current data, and you could get a fairly accurate model of what that phenotype was going to be like, that could rapidly speed up your genetic gain. And I'm not going to stand up here and say it's all about these modeled phenotypes, that through each process, these tools and information, the synthesis of this, can answer basic biological questions that we've had for quite a while. And so I just like to conclude that, in terms of increasing genetic gains, it's not new concepts. That it's just new tools that are available. And we have to kind of change our mind in terms of how we're going to incorporate that. Like for genomics, GS and GWAS, both of these depend on having high quality phenotypic data. And with phenomics, we're able to get that more and higher quality data. So when we think about how we combine these, whether it's through crop models or something to be, found yet, it's just this idea that to really synthesize biological insight from data, we need to focus on our analytical capabilities, and this is going to be a challenge. It's not easy. We're in the age of data overload, and there are no, simple solutions anymore to these problems because the complexity is so hard. And so with that, I'd just like to conclude there's always a ton of people who think on projects like this, specifically the Gore Lab at Cornell University, the USDA in Maricopa, Donald Danforth Center for doing the actual inomic analysis, helping with that. And as a final slide, if anybody's looking for a postdoc and would like to live in Tucson, Arizona, please let me know. And with that, I take any of your questions.
Thank you, Dr. Pai, for your presentation. If anyone has questions, please raise your hand, and we will have a volunteer come up to you with a microphone. A question from the webinar.
This is actually from me. Regarding the vapor pressure fluorescence system, how does that work with regards to physiological levels that we expect to see? Is it all throughout the range, or is it just limited to a certain portion?
Well, I mean, the relative humidity within inside a leaf ranges between 99 and 100% humidity, right? It's a very defined range. Because if you don't have that, the gas exchange breaks down. So with that nanoparticle sensor, it is tuned for that, pretty much those conditions.
So it was just water level, or I guess I was thinking it was other volatiles as well.
No, it's just purely water.
Purely water. Thank you.
Any more questions?
Hey, Duke.
Oh boy.
So you spent a lot of your the beginning of your presentation presenting QTL that you've mapped, and then the end you were really talking about how we're going to use various information, but you didn't really cover how you might use those QTL. Do you anticipate using those QTL in any type of way in the future?
I mean, absolutely, right? I mean, detecting a QTL is only but half the battle. It's figuring out what that, what is that signal, right? I mean, what's the gene underlying that? In terms of breeding, the QTL that we identified, those would be obvious candidates in terms of marker-assisted selection if we had the marker, we had confidence where that marker was in relative to QTL. But yes, I mean, in terms of the applied breeding, I would want to incorporate that information into, picking parents and progressing those traits in advanced germplasm. From the more basic science side, I think it goes back to, again, understanding what gives rise to those QTL, because the QTL in and of itself is not the story. I hope that answered your question, Seth.
We have another question here. Dr. McGill.
I was wondering if there's uniformity of year to year where you are actually cuts down on the ability to detect QTLs and so forth that might have an effect otherwise.
Certainly, yes. I mean, right, how much variation do you want to see in your environment? Yeah, I mean, there is that, I guess there would be that argument that because it was so uniform year to year, that maybe that hurt us. But at the same time, you have to keep in mind that our mapping population of only 95 lines, which admittedly is very, underpowered in terms of detecting QTL, it was kind of ideal. Because if we would have had too much variation with that small sample size, we might have been hosed and not seen anything. So I think in this context, yes, but I mean, going forward, obviously I'd always push for more environments in different locations.
Any more questions? 2 hands in the back of the room.
Thank you. Sorry. So going back to your data that you were talking about, and you've obviously collected gross amounts of data. And how are you, as you collaborate with across institutions and within, centers and such, how are you sharing your data, and how are you managing that data on a, obviously a lot of it?
This is going to be an embarrassing response, but when I started my postdoc, I was given 8 gigs of Excel spreadsheets. And unfortunately, that is, where we were at the time. And I spent the first 6 months of my postdoc just doing that. And so you're absolutely right. I mean, when we think about, phenotyping, it's the data question. Where is it going to live for the rest of its life? Who's going to curate it? Those are very valuable questions. So yeah, I can tell you with ours, it's sitting on my computer. If somebody was interested in using it, I'd be more than willing to share it. But I know probably not the most in-depth.
If you have USDA funding, do you have to make that data associated with your research publicly available, or is it?
Yes, although most of this project was funded by the NSF, which still mandates the same. Yeah, and so the data itself is public in the publications that are out there. However, I mean, if you contact us, we have no problems providing. I would just, it's like what repository to use. I mean, Cyverse, or what used to be iPlant, was a great avenue for hosting data and still is. I imagine at some point that's where all the phenotyping data will migrate to.
Yeah. My question is that, do you have any idea or what the causals for the temporary QTL you identified? Do you
right.
have any idea on that?
What underlies those? We There was, in terms of the QTL interval itself, when we blasted that, we found it had a few like candidate genes that looked okay, but nothing that was concrete, and certainly I wouldn't hang my hat on. So no, we haven't figured out what those are.
You think that's a variation in the gene? Because some gene may be in different stage, maybe have a different function.
Sure, right? And
also, certainly, there are RNA alternatives splicing issue. That means different transcript, the same gene. Maybe different transcript, maybe response, different temporary QTR as well. Do you think that makes sense?
I mean, absolutely. I mean, it even comes down to basic, transcription factors in terms of what's needed at the time. And so, yeah, again, I apologize. We didn't have the depth of data to answer those questions. But very interesting nonetheless, right? Because that's ultimately what we want.
Thank you.
Any more questions? Do we have any questions from the webinar? If not,
Perfect.
Let's thank
Thank
Dr.
you.
Pauly once again. Our next speaker today is Dr. Sindhuja Sankaran. Dr. Sankaran is an assistant professor in the Department of Biological System Engineering at the University of Washington State University. Something's wrong. Her research emphasis has been in agricultural automation engineering area since 2013. She is currently leading the phenomic aspects of a couple of ongoing NIFA AFRI grants, where her focus is on developing high-throughput phenotyping tools for field and post-harvest crop trait evaluation in cereal, legume, and specialty crops. Dr. Sankaran holds a BS in zoology, an MS in environmental science, an MS in environmental engineering, and a PhD in agricultural biosystems engineering. Please join me in welcoming Dr. Sindhuja Sankaran.
Thank you. Mic is on. Can you guys hear me? Yeah, I guess so. Thank you. So thank you for the student organizing committee and the sponsors for inviting me to be here to talk about phenomics. My background is ag engineering, so I come in the technological side of things, which is very different from breeding, but I'm learning a lot of things every time I'm participating in a symposium or an event. So today I'm going to talk about sensors and phenomics. Basically, I wanted it to be an educational aspect. So I'm going to talk about small things that's important for the throughput, automated image processing, as well as the benefits and limitations of the machine learning approaches. What does high throughput phenotyping mean? So we heard from Dr. Duke Pauly, and a couple years back, we met at a conference that was basically to figure out what is high throughput phenotyping. And depending on whom you talk to, the definition of high throughput phenotyping can be very different, because sometimes they are focusing on advanced techniques like X-ray CT, which is highly accurate, but probably the throughput is not there. Sometimes it can mean throughput, sometimes it can mean lower cost and rapid, sometimes it can mean automated processing of images, and sometimes it could be a combination of few things. So high throughput phenotyping, for me, it could mean more accuracy, better accuracy, and breeders are okay with relatively lower accuracy but high throughput. So because it's the relative comparison between varieties rather than absolute sometimes. So this is the overall goal for high throughput phenotyping, to figure out how do we evaluate the phenotypes or the traits. I come from Washington State, and we have around 300 different agricultural commodities. You would see there are some animal-based products like milk or aquaculture, but mostly it's plant-based. And we are top ranked in the nation for at least 10 plus crops. I'm fortunate to work on multiple crops that deals with apples, grapes, as well as a lot of legumes and wheat. In terms of breeding programs, as far as I know, there are at least 15 different breeding programs, in Washington State. And it is not just the researchers in Washington State University, but also the USDA ARS scientists. So there are centers in Washington State University, and several of those centers have also USDA ARS units and we work collaboratively on many different things. So we do have cereal grains, wheat, barley, quinoa, to name a few, legumes, chickpea, dry beans, dry peas, lentils, horticulture crops, hops, potatoes, camelina, apple, pears, cherries, and many more, including Christmas trees. And many of these breeding programs are supported by the Commodity Commission. So without their funding, their support, their involvement, the success of these breeding programs would not be possible. As far as in our lab goes, we work on several different types of sensors, which range from handheld drones to satellites. So we have the sophisticated handheld sensors that is capable of sensing visible near-infrared spectra from the leaves. We use tractors. Here is one where the sensors are mounted on a tractor. We have different system for tree crops because we want to be able to adjust the height of the sensor data capture much more than this one. And of course, the most exciting and advanced technology, which is drones, and it is growing. It's an growing era. We work on many different types of sensors. We have simple multispectral cameras with near-infrared red edge bands capable of generating generic vegetation indices like NDVI, or normalized difference vegetation index, is one. We have visible near-infrared spectroradiometer capable of capturing 350 to 2,500 nanometer. It's a state-of-the-art equipment, highly accurate. We have an imaging system corresponding to that. We have a Viz NIR, the best possible in terms of accuracy as well as the range. We have 2D LiDAR systems that are highly accurate, centimeter level accuracies, and as well as 3D systems that's lightweight, mainly for drone applications. We use quite a bit of X-ray CT system. We have a wet scope. We utilize their sensors for our applications. Simple RGB, GoPro. In addition, we also have microscopic RGB imaging, and this we use for specialized things. Sometimes we deal with very tiny seeds or tiny crops, and we find this microscopic imaging is useful in identifying the differences. And the thermal cameras, we have different versions. The latest one is the one which has both RGB and thermal integrated together. In addition to that, in our lab, we developed our own platforms. This was a platform that was inbuilt in our lab, and this was a part of the students' master thesis. The advantage of such system is it can be easily adapted. Commercial systems, if you're going to change it, you're going to void your warranty. Here you have the flexibility to add a sensor, remove the sensor, and the most important aspect, the image processing side, which is pretty rapid. In 2 minutes, it can process all the images collected during a series of 10-15 days, and it spits out the phenotype. And we have modulated systems, so each of these modules control a particular sensor which can be adapted. Each sensor comes with a software to operate that particular sensor. When you are using multiple sensors, you should be able to adapt that one software for controlling all different types of sensors. With that, I would just like to say here, all we see is in the visible range, which is 400 to 750 nanometer. But there is a range beyond visible, near infrared, X-ray, and much more that can be utilized for phenotyping applications. In our lab, we work on many different crops and with close collaboration with several breeders. And throughout this presentation, I'm going to give you several examples of it. Yield potential is important for both legumes as well as grain crops. In tree fruits, including apples and grapes, sometimes we work on disease resistance. We work on water use efficiency, again for grapes, potatoes, quinoa, water use efficiency, and these are just to name a few traits that our research lab works on. Here is an example where we are just looking at the flowering, days to flowering, and this is camelina, which is an alternative crop, and that's a seed sizing that we also do. We not just in terms of seed properties contributing to the quality of the crop, but also seed properties that influence the plant properties, better emergence, spring stand, and other things like that. The second image here is an example of water use efficiency in grapes, which is basically a normalized canopy temperature. And you can see there is no treatment effects. In 2016 winter, we had quite a bit of snow. So even if we had a water treatment, that did not play a role in inducing a stress. Although the excellent thing is still we get in relation to ship with stomatal conductance, we get excellent correlation. And that's mainly possible because we normalize all our temperature and visible near infrared data. An example of tuber, most of our potatoes goes to chip industry. So potato tuber size is an important trait that we do evaluate. And it's possible. We are currently in a stage where we are developing an app to be able to do the shape and size evaluation. Again, another example of a disease tolerance. So we have this AFRI Food Security Grant, which is a $3 million grant, which the title is over here, which our main goal was to help the students, graduate students, in several different types of breeding programs to train them to be able to use the phenomics tools in their breeding objective. So we work quite a bit. So we usually have engineering students paired with plant science students working together, understanding each other. And that process is very important if we really want to develop a high throughput phenotyping technique. So we are three cores. We have a phenomic core led by my group, genomics as well as bioinformatics. These are the breeders, including apple breeder, Dr. Kate Evans, and a range of crops. Each crop is very different in terms of cropping systems. Their goals are different. The way they grow is different. So we are learning a lot through this project. So now I'm going to talk about some basic steps of image processing before we move to the machine learning aspect. So as a part of this project, we are, just to start with, we are helping the plant science students, breeding students, basically use the NDVI cameras and extract some basic features. One is a canopy cover, the other is canopy vigor, which can be acquired with NDVI. And the major aspect over here is the throughput rather than innovation in this aspect. We are able to process 25,000 images in 2 days, which is pretty good. We have optimized all these algorithms for multiple crops, and we are able to achieve that throughput. The second example over here is an X-ray CT. So here we are using X-ray CT. In apples, there is a disorder called as bitter pit, often associated with calcium deficiency. And sometimes the bitter pit starts from within the apples rather than the surface. So we use X-ray technique mainly for production rather than breeding. It can be useful for breeding as well. But in production, what happens, they store these apples for about a year, and in a few months, when they remove it from the storage facility, this bitter pit has developed. So we wanted to develop a technique where it can still be packed, and you scan it, and you will know whether the apples have this disorder or not. And when you look at the image processing aspects, there is similarity between both the projects. And of course, here again, the throughput is important, and we can achieve a throughput in 60 to 100 seconds. We are able to process all the X-ray CT data, which is much bigger. And of course, I say without intermediate images, one thing that we learn as a technologist or a data scientist is as you process the images, it's always better to once in a while check, is my algorithm correct? Because often what happens in the process, if there is a small error in image acquisition, say that the angle of the image acquisition changed, the data can be very noisy. So it's very important once in a while to see what, to make sure whatever you're seeing is what it should be. So here I wanted to just give a sort of a brief piece of advice on different how to use image processing. The first one is know your sensors, right? So this is this is a multispectral image, and there are 3 bands. Usually a digital camera has R, G, and B, which is red, green, and blue. Here you have, instead of 1 channel, you have near infrared. Just looking at this image, you will be guessing that this is my near infrared, because plants reflect out near infrared much more than the colored, because they absorb more visible light for chlorophyll purposes, as well as they should reflect. But actually, this is the near infrared. So the first thing is you should know what spectral bands. Sometimes we have a student from plant science coming saying that they are using multispectral camera. They don't know which band is near infrared. And sometimes it's very hard to figure out. So you should know your sensor is the first advice. A field of view and resolution is important. So we work with lentils. Probably that's the tiniest crop we have worked with. So if I'm flying a drone in a field of lentils, I should know like what's the altitude is best to give me the best resolution for that particular replication. And the third one is the reliability of the sensor. And I was at Phenome conference last week, and we had a speaker say the same thing. It can be the same company, it can be the same model, but they are making revisions to their version again and again. So if you're going to change your sensor, even if the model number is same, you may be changing what data you're acquiring. And sometimes we do that because we have a group of 8 breeding students that we are borrowing the sensors for throughout the season. There's a lot of demand. So we do have multiple sensors. And one of the advice we do is make sure we have a tag in every camera. You're using the same camera every time. Because if you're going to change the camera 1 and camera 2, probably, I don't know how, we do normalize all the images. Still, sometimes there may be some differences. The second aspect is compensation. We calibrate each of our, even RGB camera images, as well as visible infrared. I cannot insist more that calibration is very important. I'm not going to talk about it, but usually each light has something called as a color temperature. So you have to, between light sources, if you want to keep your data comparable, even cloudiness, solar radiation, it's very important to calibrate. And this is just an example. Here we are looking at LED light. LED light does not have all wavelengths equal, so they have differences. But once you calibrate it, you can almost get a natural color of the root. And the same way, thermal images also, we find that calibration is really improving the quality of the data. And the third aspect is the light quality and quantity. Especially if you're doing near infrared imaging, your light source needs an infrared source. So it's a reflection that you're measuring. So your light should have a near infrared band to be able to reflect that. Imaging angle, time of the day, all those things play a role. Imaging protocol is very, very, very important if you need to automate the process. If I am able to process 25,000 images in 2 days, it is only possible if you keep the imaging protocol the same. So once in a while, based on your convenience, if you have changed things, it's going to affect your data quality. And image background is important. And sometimes this, I call it a cross-disciplinary communication. We had a prototype algorithm to do a seed size on chickpeas, and we used quite a uniform background. And a lentil student wanted to know. And I surely was expecting something like this, my lentil color would be. I did not realize they had so many different colors in lentils and even patterns within the lentil. And it's our mistake completely that this cross communication. So even if a plant science student is using a technology, they need to understand a little bit of what we are trying to do so that both of them can understand each other's problems and get the best solution. So now after this experience, we are sort of sitting with each student and establishing a protocol, because without this protocol, automation would not be possible. And finally, I talk about the oversaturation. It's very important. Sometimes you see the leaf surface shines, so it results in saturation. So if there is a saturated signal, there's no way you can correct it by image processing. I can still fix this problem up to a certain level. I can still find the relative intensities, edge of the seed to be able to still get a seed size from this image. But if my image is saturated, there's no way I can correct it. And this you have to make sure when you are in field while doing it. So basic steps of image processing. So when you take an RGB camera, there are different color space. So I said red, green, and blue. This is the standard color space. But of course, you can convert it into other color spaces like hue, saturation, and value. And there are many, many different types of color spaces that you have to choose. So you have to choose the one that best separates the background and your object. And in general, when you do background subtraction, here is an eggplant with a background, and here is a field aerial image with a background. In general, the most commonly used process for background elimination is a histogram analysis. So if you do a histogram of this image, it's divided into two groups. The result for this image is this group. That's how we eliminate the background. This is the very standard method of doing it. Sometimes we use special methods, which I'll be talking about in a minute. So once you have your processed image, you have eliminated all the noise and all the background, you extract many different types of features, and you can have color, shape, texture features, and here I'm just naming a few. Just as an example, this was a study done by Afif, my graduate student, and our goal was phanomyces infection, and we had almost thousands of images. We had 360 lines, 5 reps, healthy as well as inoculated, and just a few steps, like she had this original image, she used hue color space to separate the background, and this is how, after eliminating the background, this is the image that she gets. She extracts about 87 features just based on RGB. And I say times 3 because the region of interest can be a shoot, can be a root, or shoot and root combined. So she extracts around 261 features. So it's a lot of features, and I often say sometimes too many features can confuse the machine learning algorithm, which I'll talk in a minute. So it's like within this room, if we all take if we take your blood samples or RBC and WBC level, there is a high variability in that. So you go to a doctor you say, these are my symptoms, what do I have, right? Sometimes you can give them more, okay, my RBC level is this, WBC level. The algorithm is similar. The doctor may get confused. The same way the machine learning algorithms may also get confused. So it's very important to understand these features. So as a technologist, for me, I have to understand what is my breeder really rating in this. And of course, we did some correlation analysis and we found that the root is more focused. And aphanomyces is a root rot disease. It makes sense that the breeder is, although he or she is evaluating both roots and the shoots, they are focusing more on roots. And that's important as a technologist to understand the process. And I was talking about special methods. Instead, I wish that histogram thing always works. Sometimes it doesn't. And this is my original image. This is just by that histogram I talked about. And sometimes we use some special methods like k-means clustering. And if I have a lot of noise, I have to use it. And this is going to change my processing time. So if I can process 250 images in 20 minutes, instead of 20 minutes, it's going to take 4 hours if I'm going to incorporate a special method for background elimination. The same way here, if you see, again, the blue band is always higher than the green. We use the spectral differences. So we do blue minus green to say whether it's a potato or a background. So sometimes we have to do more than simple step. This will be still faster than this. So you have to, we are optimizing the process to see which is the best in terms of throughput. And again, here I'm talking about field. And sometimes you see this is a distortion. The plots over here are going to be much smaller and the plots at the end. And if I'm going to not correct it for distortion, it's going to again lead to a biased data. So we do distortion correction, and this is just an example of distortion correction. We do pretty well in automation. What we do is we pick the first point, last point, and equally divide it by the number of plots. So all these plots are divided so. So again, both this way as well as this way, because there are around 800 plots in this image. And if I'm going to manually go and pick out each and every plot, it's going to take forever. So there is a possibility of automating the entire process, and we do find some interesting and good results. Now I'm going to machine learning. So machine learning has been utilized quite a bit, especially in terms of big data processing. So broadly, they are classified into identification, classification, quantification, and prediction. And I will talk a little bit more detailed about it. Again, there is an overlap between each of these sessions as well. So when you talk about detection, I'm just interested whether the disease is present or absent. Okay, so it's present. It's Huang Long Bin. It's a citrus greening disease. And it's present or absent. It can be nutrient deficiency that looks very similar to Huang Long Bin. It's present or absent. If it's a healthy, it should ideally, if the machine learning algorithm is working properly, it's still absent. So there are only two categories, present or absent, when we talk about the detection. When it comes to the classification, I have different categories. I have healthy, Huang Long Bin, and citrus canker, another types of diseases. So when the machine learning algorithm is classifying different categories, then I say, okay, this is healthy, Huang Long Bin, canker. I can have other categories. I have nutrient deficiency. I can say nitrogen deficiency. I can say potassium deficiency. You can give as many classes. But this was one of the most interesting disease I ever worked with, because the symptoms are never the same. So if you grow them in the greenhouse, the symptoms are different. If you grow them in the field, they are different. And sometimes the symptoms are pretty hidden inside the canopy. So it was very challenging. Okay, when it comes to the quantification, so this is an example of hail damage in potatoes, and this I say it's a 0%, 33%, 66%, and 99%. So though this study does not summarize the machine learning aspect, but it's just to show you as an example, it's categorizing and it's no more qualitative, it's a little more quantitative, right? I'm just saying little more quantitative because the best thing that you can ever get through the machine learning is the prediction. Here I have water use efficiency, transpiration rates, tomatal conductance, and I am predicting with my visible near-infrared spectra what is the transpiration rate, what is the water use efficiency. And this I would say that's the most challenging aspect in a machine learning algorithm, to be able to predict. The reason being, biologically I can say 10.1 is same as 10.5, right? But when a machine learning algorithm is looking at 10 and 10.5, it may perceive it as different if the spectra between the two is different. So it's very different when a machine learning looks at it versus we look at it, right? So what is machine learning? So machine learning is nothing but it's learning patterns, similarities, dissimilarities from the data. And I think it's all of us have the same thing, right? When you're sending an undergraduate student or graduate student to phenotype, they are learning patterns. Okay, how do I rate this disease? How do I rate water use efficiency or senescence, right? So machine learning often deals with big data, so it's much more easier to handle big data sets with machine learning algorithms. And I do not know anything about bioinformatics or genomics, but I come across the same terms. The algorithm I'm using to handle phenomic data, sometimes it's very similar. So the methods are very similar, just that we are using it for different applications. So, and machine learning is used, sorry about that, in practical sense, like for potato industry, they scan each and every chip, right? So if they see a brown chip, they are going to, the machine learning algorithms that is connected with the camera, it's going to make that decision, this chip is not of good quality, it's not going to go to packaging. So it is used many terms in practical sense. Another good example is face recognition, right? So, or fingerprint, right? So if do I have a criminal record, right? How are they going to do without the machine learning? They are using machine learning approaches to be able to do that in a rapid throughput manner. And many tools, like nowadays, it's very much easy. MATLAB have their own machine learning algorithms. Previously, when I was a graduate student or a postdoc, I used to write 10-line code to do a 1-line code that is now, right? So now I need to develop this complex code to be able to do the machine learning. Now it's inbuilt in many of the software. And just last week, I learned that PlantCV is starting to incorporate some of those machine learning approaches as well. Again, training is important. Just like graduate students or undergraduate students going to phenotype, training is very important for machine learning approaches, right? What is a large data set? So the larger the data set, the better is your prediction or classification, whatever you may say it, right? Whatever is your application. But what is a large data set? How do you come up with large data set, right? What is the magic number? There is no magic number. It depends on the application, right? If your plants are showing high variability, you need maybe thousands and thousands of data points for the machine learning approaches to perform the same thing. If it's a simple disorder that's very consistent and you do not have much higher variability, your data size can be relative, maybe hundreds. 50s and 100s would make sense. So it depends on the variability of plants. And I think even in phenomics has a very, very, very high potential in picking out the variation within a variety, right? So sometimes you do plant height, right? The maximum number I have heard for a plant height is 3. Hardly, they will take a yardstick, at least our wheat breeders take a yardstick and measure 3 different plants. The phenomics, if you use a LiDAR, you may be able to capture much more variability within a variety. And I think that's the advantage of using the phenomics, at looking at variation within the varieties. And so this is just an example of what I spoke. So you have a training data set, you have a test data set, and you have the raw data. So in training data set, you are going to say, this is my healthy plants, this is the way it looks like, this is my disease plant, this is the way it looks like. Now you go and, I still give a test data set and I know how it performs. This is for model development. After that, ultimately, what I want to do is, I should have an unknown data set, and still my algorithm is able to perform well. That's the real challenge, right? We never have 2 seasons the same, right? I wish it was. If we had, then probably our algorithm is going to perform much better than we expect. And again, there are 2 more terms I wanted to introduce. One is a supervised learning. The one I just talked in previous example was an example of a supervised learning. Say that this is how my healthy looks like, this is how my disease looks like. Go and find out which ones are healthy and disease. That's a supervised. I'm telling it, I'm giving it a label. Sometimes we use unsupervised. Say this is all the data. I'm not going to tell you what's healthy. I'm not going to tell you what's disease. You figure out, are there groups within this data set? And here is an example that we found that 3 different groups existed within the data set. We are not telling anything that this is healthy. That's an unsupervised learning that can be also utilized for detection. I'm not going to go much over it, but there are many different models that can be used. And we can process. I'm just going for the time, I'm just going to skip some of these slides. Here is an example where I mainly talk about the apple breeding programs, where yes, the disease is an important factor. Graft compatibility is another important factor. Tree architecture is very important factor. Especially in Washington, all our architectures have a structured structure just for the ease in harvesting and other aspects. In that case, it's important to adjust the variety for tree architecture. And of course, in tree fruits, we usually have overproduction. Sometimes some orchards are not harvested because there's no place to store, right? But when it comes to quality, they are constantly trying to improve quality. McDonald's did not have apples 10, 15 years back, but now we are moving towards healthier options. So quality, you should be able to cut your apple, keep it in the table for a few minutes and not avoid browning. That's one of their objectives, right? When you look at the rose breed guidance for how you should phenotype, you see these are the 8 guidances. And in that, you say that throughput is an important one because there's a limited window in which you have to phenotype, right? And personal fatigue, repetitively doing the same thing can result in loss of reliability, morals, as well as personal sometimes, right? And if you really see high throughput phenotyping can assist in at least 5 of those 8 points. So it has a lot of potential. And I'm just going to quickly give an example on fire blight resistance in apples. This is an important disease, can result in a loss, a significant loss. It's 100 million for maintenance, for avoidance as well. The Wenatchee, Washington is called the apple capital of the world. So we have our apple breeder, Dr. Kate Evans. They are doing phenotyping, and they have already done 2 seasons. They are not going to go for 3rd season because it's pretty labor intensive and expensive as well. So what we did was we used many different types. We used visible near-infrared spectroradiometer to get and machine learning approaches. We did gator-based imaging, and we did aerial imaging. And when you inoculate one shoot in the entire tree, you're not expecting changes in the canopy level, right? You're just measuring that one branch that's inoculated. But still we wanted to see aerial, because to see if that is inducing some stress in the entire tree that we can still be able to see. We were ambitious a little bit, you can say that. Sometimes we get a good match with the ground rating, sometimes we don't. So we completely relied on the visible near-infrared spectroradiometer. Of course, what we did is imaging, and just for the time's sake, I'm just going to say, often your symptoms, which is senescing, is similar to the other ones, and you have to do some special transformation to be able to identify those symptoms. We get good classification accuracy. We were fortunate, though 2016 and 17 were completely different, we still got similar results. And these are some of the vegetation indices that we have picked out of those, because you don't want a breeder to collect again a big data set, right? You want them to quickly be able to evaluate. This is again post-harvest side. With that, I would just like to summarize by saying that hyperspectral with machine learning approaches have a lot of potential. Phenomics is an integrated approach. We are learning a lot. We as technologists or data scientists are learning a lot. And it's important that the breeder also understands the limitations and benefits so that you can optimize a process. And image processing, machine learning can learn to lead to a biased data. For example, sometimes you see 100% accuracy. I teach instrumentation and measurements. For me, there is nothing called as 100% accuracy. No sensor is 100% accurate. It can be 99.999, but never 100%, right? So it can lead, you can modify it, and sometimes we have our graduate system, students trying to see what they want to see. You can make it happen. So it's very important that you do an objective evaluation across season, across different locations. That's very important. With that, I would like to acknowledge my graduate students and a bunch of very, very, very good collaborators. Most of them are breeders from USDA and ARS as well as WSU. And whenever I have a new experiment that I want to evaluate, they are doing a lot of volunteering for planting for me, because I'm very bad at it and I can kill plants easily. So they volunteer a lot and are excited about the phenomics research. With that, that's our beautiful Palouse. I would like to thank you. And sorry, I had to skip few slides, but I'll be happy to answer questions.
Thank you, Dr. Sankaran, for your presentation. If anyone in the audience has a question for Dr. Sankaran, we will now begin a short question and answer session. If you please raise your hand, and one of our volunteers will come up to you with a microphone. For those watching online via webinar, if you would like to ask a question, please post them on the chat box. Dr. Murray.
One of the things you started off with was saying how important it was to correct things to a reference for the image, and then to standardize cameras. But I guess one of the questions I have is, in a plant breeding program where we're really trying to make comparisons between things within the same picture, how important is that when we're looking at relevant differences rather than absolute differences?
That's a very good question. And like, for example, in aerial image, right? So I have some 400 plots. If I'm capturing one image of those 400 plots and only making comparison between varieties the same day, the same time points, then probably you don't need a calibration. But we have never done that. We usually have multiple time points and multiple locations. So we think it's important to do calibration. And if you publish any sensor journal, probably they will not accept it if you do not calibrate. It's a standard method. There are also easier ways of doing, which I haven't told over here. Sometimes we use this panel, the reflectance panels. Nowadays, we get this light sensors, and the camera itself can automatically adjust based on incident light. So things are getting better now. We don't need a reference panel in the field. So things are improving. So I think, yeah, across, yes, sometimes previously we were doing that with temperatures because temperature was much difficult to calibrate than a reflectance. But nowadays we find that if we do calibrate, we are getting better data, so better correlations.
Any more questions? Dr. Zhang.
Yeah. My question is that I saw you use the cross-validation the data, right? Did you try independent validation?
So we did not use cross-validation. Most of our study, but I wanted to talk, there are two types of validating the data. One is independent validation with test data sets if you have big data. But if your data set is very small, you can still do a cross-validation, but cross-validation can be biased. So I just wanted to talk about two different ways in which we can test, but most of our data is independent test data sets.
Oh, independent. So is different accuracy, prediction accuracy different between two validation?
Yes, it will be different in most cases. So independent testing, usually the accuracies are lower than cross-validation. So an example of the difference, just for the audience sake, if you have 10 data points in an independent evaluation, I'm going to use 8 data points to develop my model and 2 data points to say that, okay, based on this model that I have trained, is it able to predict correctly? I'm saying 2, but it's 20% or 80%, or it can ratio may differ. When it's cross-validation, I have this 10 points. I use 9 points, predict 1. 9 different points, predict 1. Like that, I may do 10-fold, I may do 10 times, and then say, okay, so it's almost like it can lead to improved modeling, but the accuracies will ultimately increase because you're using the same data set again and again to develop your model.
Another thing, that means if you did one population, breeding population, you can use another population without a problem. Is that right? If your model developer use one population, let's say,
I would,
and you can use the other population.
Within the panel, if you change the panel, your results are going to differ.
And another thing, so you did this year, can the data use next year for the prediction?
That's our goal. That's our goal.
Yeah, because that's important for
breeding. That's within the panel. So in dry bean, we had, I don't remember the top of the names, Durango population or something. And we had, if it's the same panel across different season, multiple locations, the sensing can work. But if your panel is changing, sometimes the leaf structure also changes, so it may affect your results.
Any more questions for Dr. Sankaran? We have one in the front of the room.
So in the instances where you're using this to look at whether or not a disease is present, so in citrus greening and the fire blight, what is the threshold of detection? I guess I'm asking, like, can you see the symptoms before you can detect it using your...
Yes, and that brings a big difference between phenomics and precision agriculture. In precision agriculture, we are interested in early detection. So in that particular study, though a breeder would ideally wait for 6 weeks for maximum development and only evaluate at the end of season, we did data collection beyond that time point to make sure how early can we detect. And at least in that study, we see that we can find those differences much before the final stage of raking. So there's something upon inoculation, something is happening to the plant that we can pick out much earlier.
Do we have any more questions?
Yeah, you talk about the calibration before you use the sensor to collect the data. How do you calibrate the sensor? Do you use the specific traits before you use the calibration? Because in practice, in the field, you have several symptoms. For example, there has the disease, nutrition deficient. So how do you, if you calibrate it to the specific traits, but in actually you have several symptoms in the field, how do you deal with this problem?
That's a very good question. So I think you're a little confused on the two aspects. One is the calibration, right? So the calibration is to make sure the sensor is working correct, right? So that's a different that we do with all our sensors, make sure the sensor is working correct. The second aspect that you mentioned is you have a lot of things going on in the field. And when I got this position around 4.5 years back, I thought it was exciting opportunity because in precision ag, that's important where you have nutrient deficiency. Sometimes you have overspraying, so sometimes you have drought. It's very challenging given the opportunities. For breeding, I was excited because you have a very controlled experiments. If you're looking for disease resistance, you're making sure you have enough nutrients, enough irrigation if it's an irrigated, and only varying one parameter at a time, sometimes two parameters at most. So I think for breeding, it still makes sense because that variability is much more controlled. Sometimes I see weeds in the plots and it can create noise unless that weed is very different looking that I can still remove with images. But I think in practical sense on can I detect a stress? I would say probably even if based on drone based images, it can tell you something is wrong with this tree. It's sick. That's all it can tell you. What type of sickness, you have to go closer and do a little more evaluation. Still, it improves efficiency because instead of scouting the entire orchard, you're going to just focus on few points that are not right. And your sampling may also be reduced. Instead of sampling the entire group, you may just sample a few trees to identify a particular condition.
Do we have any more questions for Dr. Sankaran? If so, let's thank Dr. Sankaran once again. On behalf of the organizing committee, I encourage you to share your experiences at our event on social media platforms and tag us on all these posts. We would like to thank our sponsors once more before we adjourn for a 15-minute break. Please help yourself to coffee that is in the back of the room, and we will begin with our next speaker promptly at 10:50. For those of you who are looking for restrooms, they're at the end of the hallway on the left side. Please welcome PhD candidate in horticulture and a symposium organizing member, Nolan Bentley.
All right, well, welcome back from the break. As part of the travel scholarships and graduate student research competition, we've brought in 5 students studying plant breeding and related sciences. We selected them from among a pool of applicants to deliver rapid oral presentations. Our first presenter today is Katie Rogers. Katie is a 4th-year PhD student at the University of Florida under the guidance of Dr. Jose Chaparro in the Fruit Tree Breeding and Genetics program. Her research is focused on characterizing flowering in citrus with the goal of developing biotechnology applications to shorten the generation time in citrus and accelerate breeding efforts. Katie grew up on a citrus and cattle farm and in Wauchula, Florida, and received her bachelor's degree in horticultural sciences, plant molecular and cellular biology at the University of Florida. So please join me in welcoming Katie Rogers. The presenter today is Mahendra Bhandari. Mahendra is a 2nd-year PhD student working under Dr. Amir Ibrahim and Dr. Qingwu Xu in wheat breeding and crop stress program. He received his bachelor's degree in agriculture science from Tribhuvan University in Nepal. He completed his master's in plant, soils, and environmental science at the West Texas A&M University. His current research is in the use of high-throughput phenotyping system in wheat breeding, with a focus on disease, yield, and water stress phenotyping. So please join me in welcoming Mahendra.
Is it okay?
Yep, I got it.
Thank you for the nice introduction, Nolan. And first of all, I'd like to thank the Symposium Organizing Committee for providing me this opportunity to talk about my research. And today I'm going to talk about using aerial imagery to assess the wheat foliar disease severity. And we heard a lot about phenomics and using remote sensing in agriculture. And I will be more specific on aerial imagery and disease severity. And particularly, I'll be talking about the wheat leaf rust. Leaf rust is caused by a fungus Puccinia and is a major disease of wheat. And when the plant is infected, small brown pustules are developed on the leaf surface. This reduces the chlorophyll content of the leaf, which provides an opportunity to use remote sensing for differentiating the diseased vegetation and the healthy vegetation. You can see in the picture, in the graph over there, I'm sorry. Over here, there is a contrast in the visible and near infrared region of the electromagnetic spectrum when the sensors are exposed to the diseased leaves and the healthy canopy. So we are catching up that contrast to see if we can identify the disease and see the prevalence of the disease. And the major objective of this research is to see if we can use this imagery technology to identify the disease and evaluate the disease on different genotypes. For this, we flew DJI Phantom 4 Pro, which is relatively cheaper drone, and was mounted with RGB camera to take the RGB images. And the research was conducted in Castroville, Texas. For field data collection, we collected the data on the basis of visual ratings for infection type and disease severity. Infection type comes with the susceptibility ratings, which might be susceptible, moderately susceptible, moderately resistant, and resistant. And the severity notes were taken on a scale of 0 to 100. The infection type was later converted into the numerical value and multiplied with the severity notes to calculate the coefficient of infection. And on the image processing side, what we did is process the image to develop excess green index and band ratio. Excess green index, what it does is it highlights the green portion of the canopy in the plot. And the band ratio, it highlights the diseased portion of the canopy in the plot. And we also tried several other image classification approaches. The image over here, it is the orthomosaic image of the entire field, which is almost 20 acres of the area. And for this study, we are focusing on mainly on this corner of the field, which consists of 40 different genotypes and 3 replication. And the next picture over here, it consists of the plot boundary layers, which is used to extract the data from individual plots. With that approach, I'd like to move to the results. The results shows that both of those indices, excess green index and band ratio and the maximum likelihood classification approach, was able to identify the diseased portion over here and the healthy vegetation. The first picture over here is the normal RGB image, and it consists of, here are the little plots that are surrounded by the black boundary. Those are the plots that consist of susceptible varieties. And the plots that are surrounded by the yellow boundaries, those are the plots which are resistant varieties. So what these indices did is they were able to isolate those two different plots based on the severity of the disease in the field. And after extracting the data from the field, we could see that the excess green index and the band ratio both were highly correlated with the coefficient of infection. So to summarize, we can say that this simple technology of aerial imagery can be helpful tool for high throughput phenotyping, particularly for disease phenotyping. And what important point we need to focus is that how automated is the process of image processing and data extraction. And the take-home message from this is a simple camera and a simple drone can provide a lot of information regarding the plant behavior so that the physiologists or plant breeders can use for better study of the plants and make rational decision during the selection program. And I'd like to acknowledge my committee chairs and everyone who was involved in this project. Thank you. And I'd love to have the questions if you have any.
We do have several minutes, so if you raise your hand, we'll get this one.
So how did you had 3 approaches up there, the maximum likelihood approach, how'd that do?
It is like what we do is, I used NB for using that maximum likelihood approach. What it does is it classifies the image based on the data that we supply to the image, like supervised classification. It was better. Actually, I tried several other approaches of image classification as well, and it was relatively better than other ones.
I have a question. So when I talked to the wheat breeders, they said that sometimes with the resistance, the resistant variety does a little more yellowing, though the spots are much lower. So the severity, it tries to maintain, it looks sick, but it tries to fight out the disease much better when it comes to the resistant variety versus susceptible. So how did you see that in your...
Yeah, actually that is, I think you were talking about this earlier as well. We know something is going on in the field based on the image or based on the indices, but we are not quite sure actually. It might be because of physiological changes or maybe senescence or something like that. what we found is like those ratings were highly correlated with the visual ratings. So we still think that there is a chance of improvement, maybe being more precise for pixel selection. Let us see, say if we select the particular pixel of that brown spot, then use that particular spot as a ground truth to classify the image. Maybe that would be the better approach. And we'll be using that approach in the coming season too. It is just the RGB image view, and we are increasing the number of sensors as well, multispectral and thermal cameras. So maybe we can just stack them together and find out something.
Yeah, your correlations are very good actually. So yeah, I don't see a need for multispectral if you can get the similar data.
Yeah, that is what I wanted to focus is like, this technology is really cheap, like maybe around 2,500 or so, the Phantom 4 Pro and the camera. So multispectral will cost like $5,000 or $6,000. So maybe we can stick up in this cheaper technology and do the same job what other sensors can do.
Thank you.
All right, I think that is all the time we have. So thank you, Mahindra.
Thank you.
All right, our next presenter today is Nicholas Ace Pugh. Ace is a doctoral candidate in Dr. Bill Rooney's sorghum breeding lab. His research focuses on the utilization of remote sensing techniques for high throughput phenomics and in the implementation of these technologies into a breeding program. Please join me in welcoming Ace Pugh.
Thank you, Nolan. So as Nolan said, I'm Ace, and today I'll be presenting a project that actually is the product of a pretty large interdisciplinary effort across the university. And that project is temporal estimates of crop growth in sorghum and maize breeding enabled by unmanned aerial systems. So as I said, we have a large collaborative effort in this university to try to use UAS for various agricultural applications. And this is one of the fruits of that labor. Now, when we speak about evaluating UAS for their applications towards agriculture, there's a difference in opinion, or maybe just a difference in values between plant breeders and engineers and computer scientists. Neither viewpoint is wrong, but there's a difference in applicability depending on how you look at it. So engineers are usually going to be concerned with correlative analysis and having the highest R-square value that they can get. And that is important, but as plant breeders, we're actually usually more concerned with being able to tease out genotypic variation and implement these technologies within a breeding program. So these are just a few of the results that are presented in a recent paper on this subject. And I want to draw attention to the left side of the screen with the variation explained figure. Now, we did this study using structure from motion. That was how the UAV height estimates were derived. And we also used ground truth measurements for comparison, and those were recorded using the traditional method of a height stick. So you can see here that the variation explained between two different sorghum tests and two different maize plantings is pretty high via the UAV. Actually very similar to ground truth methods and even superior in some cases, especially early in the growth season. It's also important to draw attention to the fact that it does kind of depend whether you're measuring sorghum or maize or any other crop, what type of measurements, what portion of the point cloud that you use. So here we showed a couple of different percentiles of the point cloud data that was generated from structure from motion. So in this case, we have the max percentile and the 95th percentile. And as you can see, there is a difference between the two. But overall, the UAS are actually very good at explaining genotypic variation, and that's really important for breeders. Now, we also wanted to take this a step further and look at maybe a way that we could actually implement this technology and maybe derive new phenotypic information from it. And I want to draw attention to the right side of the screen with these high-resolution multi-temporal growth curves. Now, the sorghum growth curves A and B were generated just using a single UAV in Corpus Christi, Texas, using a Phantom 3, I believe. And C, though, the maize growth curve was actually generated using that same Phantom and two other UAVs, an EB and a Solo. So what's kind of interesting here is that we can actually derive some information that you maybe wouldn't have access to if you only took a final plant height. You can see different crossovers between genotypes that maybe occurs early in growth, which could be an estimate of plant vigor or response to stress. So right now we're just showing this kind of more in a qualitative sense, but there's probably going to be some applications soon where you try to utilize this information quantitatively as well. So future objectives for this work, I would like to kind of assess UAS for their ability to phenotype other traits of interest in sorghum. So a couple examples would be biomass yield or disease scores. And also even generate maybe curves for those traits as well, because different valuable information could be gleaned from that. And finally, I would like to also integrate these UAS height measurements into standard plant breeding studies and start to look for within plot variation. So 5 minutes isn't a lot of time, but I think I gave you a good overview of what you'll see if you come visit my poster. This project was also recently published in the Plant Phenome Journal. This is a list of most of the individuals and organizations that made this research possible. It's not an exhaustive list, though, because a lot of people worked on this project. And I would also like to thank the Symposium Organizing Committee for putting this on today and for giving me the opportunity to speak here today. And with that, I'll take a few questions.
Thank you. We have about 5 minutes for questions. Yes, Dr. Zong. There, I can. Oh, we got one coming too.
I have a little general question. Question that there's so far there are few type kind of technology for breeding, like a marker system breeding, genomic selection, right? There's certainly current traditional breeding, phenotypic selection, and now that the high throughput phenotyping. If you make a comparison to the technology, what's the advantage of high throughput phenotyping technology?
So the
over the current other technology, genomic selection, marker selection, and traditional phenotypic selection.
So what are the advantages?
Yes.
So I think the best comparison here is between high throughput phenotyping and regular traditional phenotyping. Because in that case, high throughput generally is going to be more useful if you're analyzing a large number of plots. If you want the most, I guess, accurate measurement possible, you probably would still want to use a height stick on a ground level just because you know that you're getting the absolute best. But for most breeding studies, I could see this being preferable to that method in general. As far as comparing it to things like genomics, I don't really necessarily think that they can be compared in that fashion because really I think you would utilize this in tandem with those technologies. So I think that they just complement each other really.
We have one at the back. Dr. Murray.
So since you didn't mention it, the growth curves near the end decrease. And I think many people have discussed with you why they think that is. Why do you think that is at this point? And why is that important?
So the answer to that, I guess the conservative answer would be that I don't really know. So I don't want to make declarations that aren't necessarily backed up. But so I think if I was to hypothesize, I would say that's probably due to a large number of different factors. So we know that plants senesce later in the growth cycle, and the heat can also cause droopage of plants, and there's a lot of other things, lodging. So there's a lot of answers for why you could see this decrease. Now, another possibility that would need to be investigated is that maybe plants actually have stalk shrinkage later in their growth cycle. But that would be something we would need to investigate before we make such a claim.
No, it's actually from me. So, based on this work, do you think we would be able to know which growth stage or what point in the life cycle is best for phenotyping, for vigor, or like when it's indicative of yield?
I think that's maybe an application that will be developed, because that's kind of, the growth curves kind of gets at that, and different genotypes might actually have different points where that response happens. So when you take measurements many times over the course of a season, for example, these growth curves that you see here were generated from somewhere between 15 and 20 different flights, depending on the crop. When you have that kind of information, you can really see where each genotype actually had this response. So that's kind of one of the values of having this sort of a qualitative representation of plant growth.
That's all the time we have, so let's please thank ACE.
Thank you.
Just a reminder that if you would like to hear more about any of our presenters' projects, they will be available next to their posters in the upcoming poster session. So next up, our next presenter is Suraj Sapkota. Suraj is a PhD student at the University of Georgia Institute of Plant Breeding, Genetics, and Genomics. He's working on wheat diseases, leaf stripe, and rusts for his PhD research. His research emphasizes the identification and characterization of the genetic basis of of resistance to leaf and stripe rust in soft red winter wheat cultivars. He received a Master's of Science degree from the North Dakota State University in 2015. His master's thesis was titled The Identification and Genomic Mapping of Resistance to Bacterial Leaf Streak Disease in Wheat. He received a Bachelor of Science in Agriculture from Tribhuvan University, Nepal in 2010. And with that, let's please welcome Suraj. Thank you.
Yeah. All right, thank you so much. At first, I would like to thank the organizing committee for providing this opportunity to come here and present my PhD research work. So today I'm going to talk about one of the major QTL that identified for the leaf rust disease in wheat. So wheat is a major crop worldwide, and large proportion of world population depends on wheat. So about 20% of calories that we need for human population that comes from wheat. US is a major producer of wheat, and it contributes about 10% of world production. There are about 42 states in the US they produce wheat, with Kansas and North Dakota as the leading states. Leaf rust is a major disease. There are 3 different kinds of rust: leaf rust, stripe rust, and steam rust. So leaf rust is the major one with wide distribution and most common disease. So it is also a major disease in the southeast, a major disease of wheat in the southeast, and up to 40% yield reduction is possible because of leaf rust. So one of the major problem with leaf rust is that this pathogen continuously evolves new race. So on an average, from the US, over 50 races are reported every year. So that demand us to continuously search for a novel source of resistance and novel gene. That is the reason my objective of my research is to identify QTL that we can use in the breeding program to develop leaf rust resistant wheat variety. So, so initially what I did was I evaluated the parents with these three most common rays of leaf rust, and I found that so the AGS 2000 is resistant to all the three rays. However, this Pioneer 26R61 parent is susceptible to one race and resistant to two other rays. So in this photo, you can see the reaction of the two parents with the leaf rust race one. So this is highly resistant reaction, and this one is susceptible reaction. So I chose this race and screen my real population. So I have 175 real population developed from the cross of these two parents. And this graph shows the distribution of real population to each disease category. So we see either resistant or susceptible category. There is no any intermediate reaction. So that indicates that there is a single major gene conferring resistance to leaf rust. So for the mapping purpose, I used a genetic map constructed by Howe et al. So the map includes about 2,800 markers. So the markers were SSR and SNP and dot marker. It is a kind of integrated map. So that map covered the whole genome of wheat. And from the QTL mapping work, I identified a major QTL on 2B chromosome. So this is a highly significant QTL with lot value of almost 14, and the R-square value is almost 27-28%. So I identified the QTL, but the next question is this a novel QTL? Because there are more than 70 genes identified for leaf rust. And the question is, yes, this is a new QTL. So here is the evidence. So there are two other genes reported from the 2B chromosome. So LR50 and LR58. So if we see the origin of these genes, so LR50 was originated from the wild tetraploid wheat, and LR58 was originated from bar-weight goat grass. It is highly unlikely that these genes are present in the common wheat. And another evidence is that if we see the physical location of my QTL with other two genes, we can see, so the LR58, so LR50 is located in this deletion bin of the physical map, and LR58 is located in this deletion map, and my QTL is located in this deletion bin. So this also indicates that my QTL is different than the previous one. So the conclusion of my research is, yes, I identified a QTL which is novel, and we can use that QTL in wheat breeding program to develop like leaf rust resistant wheat cultivar. With that, I would like to thank my major professor, Dr. Muhammad Marzoon, and I also would like to thank Jerry Johnson and Howe. So Howe is the person who developed this mapping population and did most of the genotypic work. I would like to thank Dan, Steve, and Ben for helping me in my research. With that, I would like to take questions if you have. Thank you so much.
Thank you, Suraj. We have 4 minutes, I believe, for questions. That wants to raise their hand. Any other questions? Yes, Dr. Klein. We'll be right there.
So this was for one race, but are you building like durable resistance by pyramiding, genes for? You mentioned using it in a marker-assisted selection breeding program, but would you need to have QTL for the other races of resistance to be able to do that as well?
Yeah, this QTL is for the race 1. So this race 1 is the most common race that we have in Georgia. I'm also screening my parent with other race from other states, and I haven't found any difference between the parent. So I'll continue doing that with almost 10 or 15 races of leaf rust. And it is highly possible that we can use this QTL and integrate with other QTL to develop a broad spectrum resistant wheat variety. Thank you.
We have a question from Dr. Murray.
You're planning on using this in plant breeding or marker-assisted selection. Is there any interest in the lab that you're in to try and identify the genetic basis of this gene, or is that just extra work? And what's your goal?
I think that's what I can do for my time right now. But the person who is going to follow up this work, he may be working on fine mapping and narrowing down the QTL region.
Okay. Any further questions? All right. Well, I guess with that, let's thank Suraj.
Thank you so much.
Our next presenter today is Anna Castro. Anna is a fifth-year molecular and environmental plant sciences, or MEPS, PhD student in the Mullet Lab. Her research focuses on the genetics and physiology of sorghum stem development. She has previously served as the president of the MEPS Students Association and the chair of the annual MEPS Symposium organizing committee. Which is a lot of work. So please, join me in thanking Anna Castro. Thank you so much for that introduction, Nolan. My research focuses on sorghum stem development and composition, and you may be asking yourselves, why should you care about sorghum stems in particular? Well, as many people have mentioned, the global population is increasing, and with that increase comes an increased demand for fuel and energy. And the continued use of fossil fuels is only exacerbating the problem of climate change. But biofuels, to some degree, close the loop of CO2 emissions and capture because plants that are used in biofuel production in turn use CO2 to fuel their own growth. The biofuels need to be grown with minimal inputs and limited competition for land and resources with other food crops. So sorghum is a particularly versatile crop, as you can see from this figure. It can be used for a lot of different uses. And it's a particularly attractive target for use as a biofuel feedstock because it is highly drought resilient, it uses light energy very efficiently, and it can be grown on somewhat marginal lands that other crops might not thrive on. Sorghum can feed into bioethanol production in several different ways, as you can see here. The grains, the juice from the stalk, and the stem and leaf material itself can all feed into bioethanol production. The ideal sorghum biofuel crop might be optimized for two or more of these traits in order to feed into bioethanol production in multiple different ways. The stem here is the main organ for accumulation of biomass and stem sugars in sorghum, but the problem is that high biomass accumulation and high stem sugar concentration may not necessarily be present in the same variety. A problem is that some varieties of sorghum selectively destroy their pith parenchyma cells, effectively hollowing out their stem, and you can see that from this progression. You can see that progression in this photo here, or in these CT scans. So the sorghum germplasm shows a lot of variation for this trait that we call parenchyma formation, and so we call this parenchyma formation. And even though this trait has been selected against in a lot of sweet and grain sorghums, we don't actually, until this point, have known much of the genetic mechanism or regulation underlying this trait. So what I've done in my research is I used the genetic resources available in sorghum to map this trait, variation for parenchyma formation, in 3 biparental populations to a locus on chromosome 6. I was then able to find map that locus to a single gene encoding a NAC transcription factor that has a single nucleotide polymorphism causing a stop codon in a truncated protein. Further expression studies that I have done show that this NAC transcription factor this is the genotype that does form parenchyma, and this is the genotype that does not form parenchyma. You can see that expression remains mostly the same as you would expect since this is non-functional at the protein level. However, these genes, several genes involved in programmed cell death, you can see are highly upregulated in the stems of genotypes that do form parenchyma compared to those that don't. A lot more work needs to be done to understand the regulation of this gene and how other internal and external influences will affect its expression and downstream parenchyma formation. But by identifying this gene, we've taken, I think, a significant step forward in designing the ideal biofuel feedstock crop. So with that, I'd like to thank my committee members and my advisor, Dr. Mullet, and all my former and current lab members, as well as the Rooney Lab, and the numerous people that have helped me in the greenhouse. And also, I'd like to thank my funding resources, the GLBRC, and the Trade Actions Chair. I'd love to take your questions. Oh, and my poster number is number 17, so I hope I can go into a little bit more detail of my research there. Questions? All right, we have 7 minutes for questions. However. Yes. Did you say you did the detail mapping when you went all the way down to single gene? I did. How'd you do this? So what I had was I had several populations that were real populations that were already available, and as soon as I started my PhD, I mapped that trait, and actually several other people in the lab had also mapped the trait. But the population I went back and mapped it in was quite a bit larger than the other 2 that it had been previously. This detail had previously been mapped in, so that just gave us a little bit more confidence. And basically, I got a little bit lucky in being able to, in the breakpoints that were available within the markers, so that I was able to narrow it down to quite a manageable region, and only had to do 1 or 2 rounds of fine mapping before I was able to identify a small enough region that had a manageable number of candidate genes. We also have whole genome sequences of these candidates, so I was able to compare those candidate genes, the sequences of those candidate genes directly. Very good. And roots can form parenchyma too. Do you think this gene is involved in root parenchyma formation? It's possible. I have not checked. We have an expression atlas within sorghum. The problem is the genotype that this expression atlas is in is a genotype that doesn't form parenchyma, which might be okay. But yeah, we think that it's possible that both stem parenchyma and root parenchyma might be, this gene might be involved in both mechanisms, and that's definitely something we'll be working on checking in my next several experiments. Good question. Does this trait make it much more prone to lodging, or? I'm not sure. I think there have been some studies on sorghum stem lodging. I don't recall if they found any significant interaction between lodging and parenchyma formation. Some people think that it's been selected against in grain sorghum by, not directly, but indirectly by selecting against the lodging. But I've read some other studies that indicate the hollower stems may be more stable. So we're not sure yet, but that'd be something interesting to look into. You have one from Dr. Mark. So the classic way to prove the function of the gene, the last step would be to do the transgenic or knockout or something like that. Do you guys mind to do that? Yeah, our lab is working on some sorghum transgenics, as I'm sure people are aware. It's transforming sorghum a little difficult. But one experiment I'd like to try to do is, since this is since the non-functional version of this gene causes the solid stems, I'd like to do basically a complementation of that gene to see if I can get those stems to also form parenchyma. Yeah. Any other questions? Any further questions? All right. Well, with that, let's thank Anna. Thank you so much. All right. So thank you to all of our rapid oral speakers today. We have now dedicated an hour today to solely poster viewing. The students that brought their posters today are studying plant breeding and other related sciences. We encourage people from across the university to participate in today's events. So we have 20, that's the newest count, 20 posters for viewing today. They are in the room behind me, behind the projection screens. This morning, the posters were judged by a panel of plant science experts. Each student presented a quick summary of their research to a subset of the judges this morning. Judges were asked to select their top 3 poster presentations, and scholarships will be awarded in the amount of $200, $150, and $100 to first, second, and third place, respectively. We ask that all students remain at their poster for the duration of the poster session. We encourage the audience members to interact with the students and get them to have a chance to explain and present their research. So I would just like to reiterate, this poster competition is only possible through the donations of our sponsors. So thank you so much. Along those lines, we also have snacks, coffee available at the back of the room. Lunch will be served at 12:30, and this is very important. So it's this hallway back here. There'll be tables back there, and you can walk through. I think we're going to start on that side. So starting at 12:30 today, we'll have a catered lunch by Jason Stone, is that right? Yeah, Jason Stone. So for those of you on the webinar, the next presentation will be from Dr. Kirti Rathore, and will begin at 1:40 p.m. Central Time. We encourage you to share your experiences on our event on our several social media platforms